Hello everyone and welcome back to Creation Myths. Today we are taking Discovery Institute to task for this video on junk DNA. So this is episode 14 in the long story short series. It's called Herbs, Pseudogenes, and Onions, and it is another entry in the Discovery Institute Tries to Defend Junk DNA saga. Now, some of you may recall that long story short episode 12 from March of 2024 was also on junk DNA, and that started a months-long back and forth between me and Discovery Institute. It involved a debate with Dr. Casey Luskin and literally months of back and forth in videos and articles and whatnot. So we are going to recall some of that stuff as we go through this new video, which was published on January 14th, 2025. Now, before we actually get into this video, let me just note that I'm not going to show the entire length. You could see in this screenshot that it's almost 14 minutes long. I'm not going to show the whole thing. I am going to show most of it, but I'm not going to show it in its entirety because there's some stuff that just isn't relevant. So right off the bat, I'm going to cut out like the first 2.5 minutes because it's a bunch of quotes from biologists about junk DNA, and I'm not interested in arguing via quote. I just don't care. If you're going to make an argument, just show me the data and make the argument. So I'm going to skip that part, and we're going to get right to the first, actual substantive argument here, which concerns the famous onion test with junk DNA. So that's what we're going to talk about first, and I'll show you this clip, then we'll come back and talk about it, and that's how we're going to do this video. We're going to show a bunch of clips. Like I said, we're going to show most of this video, and we'll intersperse it with correcting all of the errors, because let me tell you folks, I thought this was going to be a short video when I first watched this, and then I went through it a second time taking notes, and it turns out this is not going to be the short video I expected it to be. So let's get into it.
That said, there were some legitimate questions raised. Some scientists pointed to the lowly onion. If you didn't know, onions are delicious on tacos, and onions have huge genomes. Many billions of base pairs in size, 5 times the size of the human genome, in fact. Surely that entire gigantic genome can't all be functional, they say with incredulity. Actually, plants, such as onions, are often known to have a condition called polyploidy, where they can inherit extra sets of chromosomes. This can lead to the large genome size. Also, there's a positive correlation between genome size and cell volume, hinting at structural reasons for all that DNA. If you've ever looked at onion cells under a microscope in biology class, you know that onion cells can be very large, so it makes sense that they might have a lot of DNA for structural reasons.
So right off the bat, we have a lot of problems here, and to illustrate them, I'm actually going to just show a comment from my friend and co-author on a paper last year, Dr. Zach Hancock. His channel is at TalkPopGen, and I've linked it down below. Side note, if you're subscribed to me and appreciate what I do, give Dr. Zach Hancock a subscription because you're going to like what he does too. So I'm going to read this comment in its entirety and then just point out some of the high notes here. So he commented on this video. Oh, onions have so much extra DNA for cell structure, huh? Interesting. I wonder why that's only true for some onions. Also, most onions are diploids, not polyploids. Genome sizes range in onions from 7 to 32 gigabases. Why does one species of onion need 5 times more DNA for structural reasons than another? This is also not even just about plants. Krill have a 48 gigabase genome. Their structural integrity is due to chitin, so there's no structural reason to have so much DNA, nor is it due to polyploidy, as it's also a diploid. The biggest failure of the functionalist hypothesis is its complete inability to explain the immense variation in genome size across the tree of life. If the genome is entirely or mostly functional, why does an amoeba need so much more than a human? Or a pufferfish so much less than a carp? These are all great points, and I want to point out 3 things here, 2 of which come directly from Zach's comment. First is that most onions are not polyploid, so while it's true that many plants might be tetraploid or hexaploid or even octoploid, that's not the case for most onions, which of course is the basis for the onion problem. So the excuse that is provided in this video doesn't actually address the problem. Additionally, the same problem exists for animals. You can call it the onion problem, or you can call it the fish problem, or the krill problem. It's the same problem. And for animals, 1, we're intolerant of polyploidy, unlike plants. And 2, as Zach said, for example, for krill, it's not a structure thing because that's what the chitin is for in those organisms. And then third, I just want to point this out. Discovery Institute is doing a little correlation causation switcheroo right here by saying that the function of all that DNA is to stabilize the very large cells. They might be putting the cart before the horse because they provide no evidence that it's the case. They present, as a creationist might say, a just so story. Why can't it be the other way around? Why can't it be that these cells just happen to have a lot of DNA and that results in the cells being larger volume? Discovery Institute has made no effort to distinguish between those 2 possibilities, so that's that as far as I'm concerned.
Many evolutionists cite things called pseudogenes and ERVs as proof of evolution in junk DNA. Pseudogenes are sections of DNA that look like functional genes, but are thought to have a mutation that stops them from working. In other words, they're junk. But even if that was the case, pseudogenes wouldn't necessarily refute intelligent design any more than a rusty, broken down car refutes Ford. No one thinks the broken state of a thing proves it wasn't originally designed. But it turns out many pseudogenes aren't like rusty, broken cars. They're actually functional and they work just like they're supposed to. Kind of like a turn signal. They work, people just don't use them. Pseudogenes can be normal regulatory elements of our genome, which are designed to look like protein-coding genes because of how they need to function. Evolutionary scientists totally missed this key function for pseudogenes. This paper cautioned that pseudogene function is prematurely dismissed due to dogma. Here's a famous example. Brown University biologist and ID critic Kenneth Miller testified in a court trial that the human beta-globin pseudogene is broken because it has molecular errors that render the gene non-functional, indicating humans share a common ancestor with apes. 2 years later, leading evolution advocate Eugenie Scott claimed this pseudogene isn't going to do diddly. It's just going to sit there and not do a thing. But in 2013, a study proved them both wrong. This pseudogene is functional after all, and a 2021 study actually found that it is essential and has indispensability for human red blood cell formation. The reality is that just like other claims of junk DNA, this one falls apart since pseudogenes do have important functions like these papers detail.
So now we're getting into the meaty part of this video on pseudogenes and ERVs. Starting with pseudogenes, it's true that some pseudogenes are functional. We've documented function directly in maybe a dozen or 2 pseudogenes in the human genome. However, that's out of 15 to 20,000 pseudogenes in the human genome. We've directly documented function in, fewer than 20, give or take. If you want to say that most pseudogenes are functional, there's a bunch of questions you, Discovery Institute, need to be able to answer. Here they are. What percent of pseudogenes in the genome, not even transcribed, what percent of pseudogenes in the genome interact with regulatory proteins that affect protein-coding genes or other functional elements of the genome? What percent of pseudogenes are transcribed, and what percent of those transcripts survive more than just a few seconds within the nucleus of the cell? What percent of those transcripts interact with the genome in a sequence-specific fashion, as is proposed where pseudogenes are regulating gene expression in some way in the nucleus? What percent are processed and exported from the nucleus so that they can have a function in the cytoplasm? Of those, what percent interact with mRNA in a sequence-specific fashion? I'm actually asking all of these questions. These are real questions that I would really like for somebody who is on the pro-junk DNA side of things. Doesn't have to be Discovery Institute, but somebody who says most of those pseudogenes in the human genome are functional. You need to answer these questions for the vast majority of pseudogenes in order to make that claim. If you're going to say almost all of them are functional, you better have answers to these questions. And obviously in this video, we got no answers to any of these very important questions when it comes to are pseudogenes functional. So now we're going to change gears and talk about ERVs for a little bit. Let's take a look at the first of those parts.
Well, what about ERVs? Endogenous retroviruses are another class of junk DNA, commonly cited in favor of evolution. These are claimed to be ancient viruses that infected some common ancestor of both apes and humans, because now they're found in exactly the same spot of DNA in both. And, as the argument goes, the likelihood of these viruses being inserted by chance into the same place in the genomes of different species is very low. So, common descent is the better explanation. Intelligent design scientists agree they didn't arise by chance. That'd be silly. Like plagiarism, if two students have the exact same set of words in a section of their papers, the chances of those words being put together in that particular order independently or by chance are so low that an outside observer can rightfully suspect common ancestry, or plagiarism. The two students copied from a common source. But the argument for plagiarism falls apart if, say, the teacher had structured the assignment to require the insertion of a certain quote into the opening paragraphs of each student's paper. Then there's a reasonable explanation why those words appear at the beginning of both papers. The insertion of the duplicate language is neither chance nor plagiarism, but a result of the way the assignment was intelligently designed by the teacher.
Okay, so first we get a common design argument here. I've addressed this at length. Other people have addressed this at length. I'm going to link these videos down below. I covered this in Creation Myth, Common Design, and more recently in Creation Myth, Kinds, which is along the same vein, but a more specific kind of version of that case for common ancestry rather than design. So I'm going to link those two videos down below. You can check those out. I'm not going to rehash the argument here because I did a comprehensive job there. No to common design. That argument holds no water, and you can check those out for why that's the case.
Likewise, the argument for common ancestry falls apart if the ERVs aren't randomly inserted. If these ERVs prefer certain spots in the DNA, that could explain the similarities without appealing to chance or common descent.
Okay, so first I want to point out the interesting thing that's going on here, where Discovery Institute is disputing common ancestry, and usually they don't like to touch that one, and in fact a bunch of people at Discovery Institute are more or less okay with common ancestry. I'm thinking about Michael Behe, for example. So I just think it's interesting, and I don't have anything else to say about it, but it's just interesting that Discovery Institute is more overtly aligning themselves with kind of more clear creationist arguments. Typically, as kind of the intelligent design people, they try to distance themselves from the other flavors of creationism, but by coming after common ancestry here, they're really aligning themselves with organizations like AIG and CMI, for example, and it's just interesting that that's kind of a new thing for Discovery Institute. That's not something that they've always done. More importantly for what we're doing here today, there are additional problems with the arguments they're making. So let's start with hotspots. The text on the screen here reads, if these ERVs prefer certain spots in the DNA, that could explain the similarities without appealing to chance or common descent. And the argument here is that you have endogenous retroviruses, viral DNA, it's gotten inserted in the human genome and then inactivated, and you get this dead virus called an endogenous retrovirus. Now, when you look at, for example, humans, chimps, gorillas, and other apes, you find endogenous retroviruses in the same place in the genome, like in the exact same place, and the pattern of similarity is a nested hierarchical pattern across, for example, all of the great apes, including humans. Discovery Institute is trying to attribute that to hotspots, saying there are sites where the viruses preferentially insert. And while I have this on screen, I want you to note that teeny little text in the top, because that's where they're actually making their argument here, trying to preempt the counter argument And I want to highlight that because, man, this is really out there. So let's go right to this. This is what it reads across the top of that slide. Quote, evolutionists might claim that the hotspots are fairly general and not precise, but ask them how they know that's true, and you probably won't get a good answer. For all we know, hotspot insertion sites could be highly precise. So, I'm an evolutionist. Let's ask me how I know it's true that hotspots are not precise. How do we know that's true? Well, we know that's true because there are a number of viruses in the present that infect humans and inject their DNA into our genome. And while those viruses do have hotspots that they tend to land in certain regions of the genome, they are not precise enough to insert at the exact same site. And they will, of course, land outside the hotspots on occasion too. But there's a lot of variation within the hotspots, which can be quite large regions of the genome. So, for example, we can look at HIV, Epstein-Barr virus, HPV, and looking at HPV specifically, 3 to 10% of cases, the literature I could find was a little ambiguous, but as far as I could tell, 3 to 10% of HPV cases are associated with cervical cancer. If the integration into the human genome was specifically targeted, it would be close to 100% of the cases, because the virus would land repeatedly in the place where it affects the genes that downstream could result in the development of cancer. The same thing is true for HIV, the same thing is true for Epstein-Barr. These are viruses that are associated in some fraction of cases with cancer due to where the virus inserts. But if the virus reliably inserted at that cancer-associated site, it would be almost entirely associated with cancer instead of just a few cases, a single-digit percentage of cases. So this hotspots attempt does not work to explain the pattern of similarity in these endogenous retroviral sequences.
Even more important than that, though, ERVs show widespread evidence of function. They're not all simply dead ancient viruses, though some may be. They have important roles, particularly with gene regulation, developmental functions, and they're even involved in immune-related functions to repel viral infections. In fact, many ERVs may look like viral DNA because they have functions related to fighting viral infections. This is important. If the DNA is functional, then it provides no special evidence for common ancestry.
There is an even bigger problem with the ERV argument, and it is all those functions that we just heard. Gene regulation, developmental functions, immune response related to fighting viral infections was the specific wording. What do all of these functions for endogenous retroviruses have in common? They require sequence specificity. For example, if an endogenous retrovirus sequence is too different from the sequence of the target actually make you sick virus when it invades your cells, then it's not going to do its immune job, either through direct interaction or some other way. There's going to be a specific sequence that mediates that interaction. And if you don't maintain sequence specificity through purifying selection, then it's not doing that job. Because once the sequence diverges too much from the target, it simply won't work, right? So that's true for regulation, it's true for regulating development specifically, it's true for fighting viral infections. You need sequence specificity. So if there's no purifying selection, those functions are not happening, period. So now let's move on to Discovery Institute's attempt to address a completely different argument. And before we get into this, I will tell you that it's possible this one is worse than what we've talked about already.
Other scientists appeal to blatantly circular arguments. For example, consider yourself compared to, say, this mouse. Both of you have DNA, so far so good, and much of that DNA is the same. This makes sense because both of you are mammals, so you'd expect to share certain traits with each other. Other parts of your DNA, however, are different. This also makes sense because, well, different things are different. But to some evolutionists, those differences are proof that junk DNA exists, regardless of what ENCODE or others have to say.
Okay, first, straw man alert. Here's the wording from the video. Quote, to some evolutionists, those differences are proof that junk DNA exists. Now, this is wrong in two ways. In the narrow way, it's an oversimplification of what the argument would be. The actual argument, for being a little bit more precise here, would be, for example, if you take humans and mice, and you see that there's a small percentage of that genome that in those two lineages is experiencing purifying selection, right? And for humans and mice, the actual number for that is about 2.2%, right? And that would be saying, okay, it's that common area that's experienced purifying selection in that entire evolutionary history. That's functional, and the rest is proof that junk DNA exists, right? That's the more technically correct way to make this argument. Here's the problem. Nobody's making that argument, right? So the wording is an oversimplification, but also this is just straight up a straw man. No one's actually making that argument, as we're going to see in a few minutes. Let's continue.
So the argument goes like this. You and this mouse are different because of accumulated mutations. Those mutations will most easily accumulate in non-functional regions.
So evolution predicts that these different regions must be junk DNA.
Okay, look, again, this is a straw man argument. Nobody makes this argument. So let's go through it. First, you and this mouse are different because of accumulated mutations. True, yes, I agree. Those mutations will most easily accumulate in non-functional regions. Conditionally, yeah, I think that's pretty okay. I would say non-constrained regions, right? Regions not under purifying selection, but non-functional regions are more likely to be unconstrained compared to functional regions, so I think the second one's okay. And then we get to the third one. So evolution predicts these different regions must be junk DNA. No, that's wrong. That is a straw man, and this is frustrating because I go out of my way. Personally, when I'm doing this, I go out of my way to make sure that I am fairly and accurately representing creationist positions. If you go through my videos, you'll see I start most of them with, here's the quote, here's the paper, here's the example of a creationist making the argument that I am addressing. Why can't creationists do that same thing? Why can't creationists, especially if they're so confident that they're correct, why can't they extend that same courtesy to their opponents? Why can't they make an accurate version of the argument they are supposedly addressing in this video? As we're going to see in a minute, they pull up a paper related to this. So why can't they just make the argument correctly when they have a source for the argument? I don't know, but it's very frustrating. Let's continue.
A 2014 paper used this reasoning to claim that only 8.2% of human DNA is functional, because that's the amount of DNA that's similar between humans and other mammals.
Okay, there are so many problems with how that paper was described and how the findings were represented and what the implications are. So let's just break this down in a little bit of detail. All right, so what those authors described was 2.2% of the genome is conserved across mice and humans, which is to say you've got 2.2% where you've got purifying selection in humans and also in mice, and it's the same regions from their common ancestor, purifying selection. Now, within the human genome specifically, we have 8.2% of the genome is constrained, which is to say 8.2% of the human genome experiences purifying selection. Therefore, a maximum of 8.2% of the human genome has a function that requires sequence specificity. Because if there's no purifying selection, then the sequence in a region doesn't matter. It's not doing anything important enough for natural selection to be able to keep that sequence the same. The authors explained this. Like, you could just read the abstract and it's right there, and they go into way more detail later in the paper. But I'm just going to read this line that I've highlighted here in the abstract. Quote, "We estimate that 8.2% of the human genome is presently subject to negative selection and thus likely to be functional, while only 2.2% has maintained constraint in both human and mouse since these species diverged." The authors are very clear about the implications and the findings of their results in this paper and what conclusions they think we should draw. I think that 8.2% might be a little bit low, Because I think, and I think it's reasonable to think this, there are unconstrained sequences that are still functional by a reasonable standard of function. So I think that 8.2% might be a little bit low, and I think documenting purifying selection, or as the authors say, negative selection, is not the only test for function, but it is a definitive test for functions requiring sequence specificity. Now let's compare that highlighted bit right there to the representation of these findings in this video from Discovery Institute. There's the abstract, and here's the representation. So evolution predicts these different regions must be junk DNA, must be junk DNA. The regions outside the 2.2% shared constrained DNA between humans and mice. Now look, the authors said 8.2%, probably functional because it's constrained in humans. That's the standard they're using, constrained within a single species, not constrained since it diverged with a related species with the same constrained region. If the authors were doing what Discovery Institute is claiming they're doing, they would have used the 2.2% number for function instead of the 8.2% number. I don't know if the person who scripted this video read this article and didn't understand it, didn't parse it correctly. Maybe they just read the title and didn't even read the abstract. I don't know. But the list of possible explanations for why this discrepancy exists is a relatively short list, and none of them are good for Discovery Institute.
Others rely on a junk of the gaps argument, claiming that until we find function in a gene, we should assume it's junk. But this argument is defeated by the trend line of the data. We're finding function practically everywhere we look. Regardless, they have faith that maybe the next gene right around the corner will be junk this time.
Okay, look. I'm now starting to get a little bit frustrated because this is a very simple thing. That section, using this graph, that's not what that graph shows. The narrator says, quote, others rely on a junk of the gaps argument claiming, quote, until we find function in a gene, we should assume it's junk. But this argument is defeated by the trend line of the data. We're finding function practically everywhere we look. And you can see right on screen, this is the piece they cited. Now, this is not a primary publication. It's more of a review. But this graph appears in the paper that is referenced at the top. So I pulled it up to see exactly what this graph is showing, because you can see that that red line, right, other genomic elements. Now, the blue one is protein coding genes, and we got about 20,000 of those, give or take, and we've known that for a while. But it's these other genomic elements that just keep increasing, almost exponentially. But here's the thing, this is not showing function. This is showing other genomic elements discovered. That's what the y-axis is here. It's just discovered. Have we documented 130,000 functional, ERVs and pseudogenes and transposons? No. We've discovered 130,000 other genomic elements. That includes things we knew were functional, like promoters. It also includes, and I'm not joking, you can go to the paper and look this up, it also includes SNPs, single nucleotide polymorphisms. Is Discovery Institute making the completely off-the-wall claim that all single nucleotide polymorphisms are functional? I don't think they are. I don't think any creationist would make that argument. But that's the argument you need to make in order to claim that this line is indicating an exponential increase in known functional elements in the human genome. That's not what it's showing. It's just showing how many other genomic elements we've documented. There's no charitable way to read this, what I'm showing you on screen right here. Either the person who scripted this video did not read the paper that they are referencing, or they did, and they're deliberately misrepresenting what this graph shows. I don't know what the answer is, but it's one of those two things. All right, let's get to just one last thing here, and then we will wrap this up with a bunch of concluding thoughts.
Like many others, University of Houston biologist Dan Grauer realized that a lot was riding on junk DNA. He too understood that you can tell a lot about a theory by the predictions it makes. He said this: if the human genome is indeed devoid of junk DNA, as implied by the ENCODE project, then a long, undirected evolutionary process cannot explain the human genome. If, on the other hand, organisms are designed, then all DNA, or as much as possible, is expected to exhibit function. If ENCODE is right, then evolution is wrong.
Folks, if ENCODE is right, then evolution is wrong on your junk DNA bingo card, congratulations, you can fill in that square. Because of course we get the Dan Grauer, if ENCODE is right, then evolution is wrong quote. This is from 2013. I don't know what else to say to this. I've already covered this specifically. I made a video on this. I already covered it. I don't know why anybody is still making this argument. I know tons of creationists do. Discovery Institute does. Sal Cordova does. I don't know why. We all know what this means at this point, and it doesn't mean what creationists say it means. I'm not going to go through the whole thing. That video's linked down below. Feel free to watch it if you want. Now, on Evolution News, the Discovery Institute Center for Science and Culture blog, this video got not one but two promoting articles in the span of just a few days. So we have one from David Klinghoffer, new long story short, ERVs, Pseudogenes, and Onions, and Dr. Casey Luskin, new long story short video addresses challenges from junk DNA defenders. Neither of these articles add anything to what was in the video, but I just think it's notable that there were two of them within just a couple days. Discovery Institute was really pushing this new video. Before we wrap this up, let's just end with a few things that were curiously not mentioned in this video. First, there was no mention of Dr. Casey Luskin's admission to me last year in our debate that we have documented functions for less than half the human genome. Discovery Institute went out of their way last year to make it seem like what Dr. Luskin admitted had been their position all along. Now, we had a long back and forth on this where I said that Dr. Luskin made this admission and made them change their argument because afterwards they basically said, no, we've been saying that the whole time, which they clearly haven't, which I pointed out in this video. Discovery Institute is trying to gaslight you. I think it's notable that what is essentially the follow-up video on last year's discussion made no mention of this admission, and in fact, Discovery Institute seems to have reverted to its pre-admission position that function has been documented for virtually the entire human genome. That was one of my options in terms of what Discovery Institute might do. One of the potential outcomes was they just went back to their old position, like that debate and Dr. Luskin's admission never happened. And that seems to be what's happening here. So we'll see if that changes again or not. Additionally, not mentioned in this video was the RNA frequency problem. Basically, if a part of the genome is going to get transcribed to RNA, and that RNA is going to do something, there has to be enough of that RNA in the cell to do the thing it's going to do. How much transcription for these elements? Now, this came up quite a bit in that back and forth last year, and I addressed it specifically in this video looking up the primary data that went into one of the papers on this question. Dr. Luskin says he will address this separately. In the article I showed just a minute ago, in which he promoted this video, he wrote, quote, "In an upcoming post, I'll review an important rebuttal we didn't have time to address in the video. The claim that ENCODE only found low copy number RNA transcripts and that these could not have been functional." So I eagerly anticipate reading this follow-up piece. I want to see exactly how Dr. Luskin addresses those data. Dr. Luskin, if you see this, do recall that I looked up the primary data cited in the relevant ENCODE papers. The links and my discussion of it are in that video, so you can look up those data directly and go from there. And most importantly, the elephant in the room, not mentioned, purifying selection. And let's briefly go through why this matters so much. Only 8% of the human genome experiences purifying selection, which is to say selection that keeps a sequence the same. Over half of the human genome is transposons, ERVs, and pseudogenes, which means if the vast majority of the genome is functional, then transposons, ERVs, and pseudogenes better be functional. However, virtually none of those elements experience purifying selection, which means they aren't doing specific regulatory, developmental, or immune functions, all of which require sequence specificity. So what are their sequence-independent functions, and what is the evidence for those specific functions? To date, Discovery Institute has not addressed this question at all. Until they do, they have nothing. They're claiming a bunch of functions for most of the genome when most of those sequences can't be doing those functions because they don't have purifying selection. The functions require sequence specificity. The supposedly functional DNA doesn't have sequence specificity. It's not doing those functions. It's not more complicated than that, so you need to come up with functions that are independent of sequence. Tell us what those are and what the evidence is for those functions. So in conclusion, this video is full of straw man arguments, old talking points that have already been addressed. I mean, how many had I already specifically addressed on video, where I can just say, I already talked about this, go here, it's done, rather than have to explain it again. There's nothing new here. We also have incorrect interpretations of published data, and most importantly, questions not answered. The onion problem, specific numbers around pseudogenes that you need to be able to produce in order to show that they're functional, and of course, addressing the lack of purifying selection for the vast majority of the genome. Still no answers to those very specific questions that I and others have been asking of junk DNA proponents for a long time. Still no answers. So do better, Discovery Institute. We just had a months-long conversation about junk DNA. And I know this isn't about me. I get that. But I raised specific arguments, some of which were kind of new, some of which are familiar arguments that you just should have an answer to, and we don't have answers to these questions. I would have hoped that after months have gone by and you put out junk DNA content, some of it would have addressed what was left over from last year's conversation on the topic. That does not seem to be the case. So I do hope and recommend that from here you'll be addressing the arguments that people are actually making instead of the straw man arguments that we see in this video, and of course, an effort to go after the arguments that were not mentioned in this video. And that is all I have to say about that long story short video on junk DNA. Thank you all for watching. Please remember to hit the like button, subscribe, leave a comment, and do consider becoming a channel member. If you become a member at any level, you'll get immediate access to pre-recorded videos instead of having to wait for the public releases. Thank you in particular to my members. So that's it. Stay safe, stay healthy, and as always, don't get fooled.